Bismillahirrahmanirrahim. I am Dr. Zahra Noreen, course instructor of Plant Physiology 1, BOTN 3119. Today we will talk about electron transport pathways in chloroplast membranes, in other words, Z-scheme. Introduction. Between 1940s and the early 1960s, three seminal findings formed the basis of our current understanding of the coordinated operation of the two photosystems in the chloroplast electron transport chain. The first discovery was of the red drop, where the efficiency of photosynthesis was found to decrease dramatically at wavelength greater than 680 nanometer. Though there was still substantial amount of light absorbed by chlorophyll, in effect, light of wavelengths longer than 680 nanometer appeared ineffective in photosynthesis, regardless of absorption by the photosynthetic pigment. Questions raised by the red drop were answered by the second observation, the Emerson enhancement effect. When red and far-red light were used in combination, the rate of photosynthesis was greater than the sum of the rates achieved by using only red or only far-red light. The third finding revealed that red and far-red wavelengths had an antagonistic effect on the oxidation and reduction of cytochromes in the electron transport chain. Red light causing reduction of cytochromes and far red causing their oxidation. So these three findings form the basis of Z scheme, in other words, zigzag. The Z word is from zigzag, a model of the chloroplast electron transport chain that takes into account the different pigments and reaction center composition of the two photosystems. Photosystem 1, with its longer wavelength reaction center, P700, is more efficient in far-red light. Photosystem 2, however, operates more efficiently in red light because of its relatively shorter wavelength reaction center, P680. For photosynthesis to occur with optimal efficiency, the two photosystems must cooperate. Electron carriers organized in Z-scheme. Photons excite the specialized chlorophyll of the reaction center P680 for PS2 and P700 for PS1, and an electron is ejected. The electron then passes through a series of electron carriers and eventually reduces P700 for electron from PS2 or NADP for electrons from PS1. Much of the following discussion describes the journey of these electrons and the nature of their carriers. Almost all the chemical processes that make up the light reactions of photosynthesis are carried out by four major protein complexes: photosystem 2, the cytochrome B6F complex, photosystem 1, and ATP synthase. These four integral membrane complexes are vectorially oriented in the thylakoid membrane to function as follows, as shown in the figure. The figure is showing the transfer of electron and protons in the thylakoid membrane is carried out vectorially by four protein complexes PS2, cytochrome B6F, PS1, and ATP synthase. Water is oxidized and protons are released in the lumen by PS2. PS1 reduce NADP to NADPH in the stroma via the action of the ferredoxin. And the flavoprotein ferredoxin NADP reductase, in other words FNR. Protons are also transported into the lumen by the action of cytochrome B6F complex and contribute to the electrochemical proton gradient PMF. These protons must then diffuse to the ATP synthase enzyme, where their diffusion down to the electrochemical potential gradient is utilized to synthesize ATP in the stroma. Reduced plastoquinone and plastocyanin. Both these proteins are used for the transfer of electron to cytochrome B6F and pigment system 1 respectively. Plastoquinone is utilized to transfer the electron to cytochrome B6F, while plastocyanin is utilized to transfer the electron to PS1. Both these proteins are mobile in nature. Mechanism of non-cyclic electron transport chain in chloroplast. We can summarize it into following steps. Photosystem 2 oxidize water to oxygen in the thylakoid lumen and in the process release proton into the lumen. Cytochrome B6F receive electron from PS2 and delivers them to PS1. It also transports additional proton into the lumen from the stroma. PS1 reduces NADP to NADPH in the stroma by the action of ferredoxin and flavoprotein ferredoxin, NADP reductase, in other words FNR. And finally, the ATP synthase produces ATP as protons diffuse back through it from the lumen to the stroma. Here is the detail of the Z scheme for oxygen evolving photosynthetic organism. The redox carriers are placed at their midpoint redox potential at pH 7. The vertical rows representing photon absorption by reaction center chlorophyll, P60 for photosystem 2 and P700 for photosystem 1. The excited PS2 oxidized by the light is re-reduced by YZ, which is an electron donor, and it receives the electron from the splitting of water or the oxidation of water molecule. On the reducing side of the PS2, to the right of the arrow joining plastocyanin. Plastocyanin is the mobile protein, is a soluble protein, which in turn reduce the P700 or oxidized P700, the acceptor of the electron from P700, A0, is thought to be a chlorophyll, and to next acceptor quinone, A1, is a quinone molecule. A series of membrane-bounded iron-sulfur proteins then transfer the electron to the soluble ferredoxin, and then ferredoxin transfer these electrons to the flavoprotein ferredoxin NADP reductase enzyme, and which is utilized to form NADPH from NADP, or which reduce the NADP to NADPH, which is then used in the Calvin cycle to reduce carbon dioxide. The dashed line indicates the cyclic electron flow around the PS1. If electrons are not given to the FNR, the ferredoxin return it to the cytochrome B6F complex, where again it is travel through the PS1 and reach to the ferredoxin, and in this form, cyclic manner, more ATP are produced because cytochrome B6F complex is, you can say, attached with the removal of hydrogen ion into the lumen, and thus causing the accumulation of hydrogen ion into the lumen, causing a proton gradient, which is utilized by the ATP synthase and then utilized for the formation of ATP. In the Z scheme, Pheophytin and two quinones accept electron from photosystem 2. Evidence indicated that Pheophytin acts as an early acceptor in the photosystem 2, followed by a complex of two plastoquinones in close proximity to an iron atom. Pheophytin is a chlorophyll molecule in which the central magnesium atom has been replaced by two hydrogen atoms. Means you can say that pheophytin is a chlorophyll molecule but without magnesium. This chemical change gives pheophytin chemical and spectral properties that are slightly different from those of the chlorophyll. Two plastoquinones are bound to the reaction center and receive electrons from pheophytin in a sequential fashion. Transfer of 2 electron to QB reduces it to QB minus 2, and the reduced QB minus 2 takes 2 proton from the stroma side of the medium, yielding a fully reduced plastohydroquinone, which is shown in the next figure. The plastohydroquinone then dissociates from the reaction center complex and enter the hydrocarbon portion of the membrane because it is mobile in nature, where it in turn transfers its electron to the cytochrome B6F complex. Unlike the large protein complexes of the thylakoid membrane, hydroquinone is a small, non-polar molecule that diffuses readily in the non-polar core of the membrane bilayer. Here is the structure of plastoquinone. You can see here a ring structure along with a side chain, along with this unit, 9 times this unit and hydrogen. Now I will tell you about the cycle of the quinone. When quinone receives 1 electron, it will be converted into a simple quinone into plastosemiquinone, which is represented here by a dot and a minus sign. And overall, it has an oxidation state, minus oxidation state. By receiving another electron, it will be converted into plastohydroquinone. But while doing this, it also takes the 2 hydrogen from the stroma and attached itself on this side, one here and one here, and convert it into plastohydroquinone. After releasing these 2 hydrogen, this will again convert it into simple quinone, which is again able to receive 1 electron and 2 electrons and again 2 proton and convert it again into plastohydroquinone. And in this way, the cyclic fashion of conversion of quinone to plastohydroquinone will take place. And what is the benefit of this cycle? That in each turn of 2 electron, 2 hydrogen ion will be transferred from the stroma to the lumen of the thylakoid. Now the role of another major complex of Z-scheme, cytochrome B6F complex, that also transports protons. The cytochrome B6F complex is a large multi-subunit protein with several prosthetic groups. It contains 2 B-type, which is also known as cytochrome B6 heme, and 1 C-type heme, which is also known as cytochrome F. In C-type cytochromes, the heme is covalently attached to the peptide, but in B-type cytochrome, the chemically similar protoheme group is present, but it is not covalently attached. In addition, the complex contains a risk iron sulfur protein named for the scientist who firstly discovered it, in which the 2 iron atoms are bridged by 2 sulfur atoms. The structures of the cytochrome F and the related cytochrome BC1 complex, which is found in the mitochondria, have been determined and suggest a mechanism for electron and proton flow. The precise way by which the electron and proton flow through the cytochrome B6F complex is not fully understood, but a mechanism known as the Q cycle accounts for the most of the observation. In Q cycle, the plastohydroquinone is oxidized and one of the 2 electron is passed along the linear electron transport chain towards the photosystem 1, while the other electron goes through a cyclic process that increases the number of proton pumped across the membrane. In the linear electron transport chain, the oxidized risk protein accepts an electron from plastohydroquinone and transfer it to cytochrome F. Cytochrome F then transfer an electron to the blue colored copper protein plastocyanin, which in turn reduces oxidized P700 of PS1. Here is the structure of the prosthetic group of B and C type cytochrome. The protoheme group, also called the porphyrin 9, is found in B type cytochrome, and the heme C group is C type. In the C type cytochrome is present. The heme C is covalently attached to a protein by a thio ether linkage. This is the thioether linkage, two thioether linkage, and as attached to this protein, and the protoheme group is not covalently attached to the protein. The iron is in the +2 oxidation state in reduced cytochrome, and in case of oxidized cytochrome, its oxidation state will be +3. In the cyclic part of the process, the plasto semiquinone transfers its electron to one of the B-type hemes, releasing both of its proton to the luminal side of the membrane. The B-type heme transfers its electron through the second B-type heme to an oxidized quinone molecule, reducing it to the semiquinone form near the stromal surface of the complex. Another similar sequence of electron flow fully reduce the plastoquinone, and which picks up proton from the stromal side of the membrane and is released from the B6F complex as plastohydroquinone. The net result of these two turnover of the complex is that two electrons are transferred to P700, two plastohydroquinone are oxidized to the quinone form, and one oxidized plastoquinone is reduced to the hydroquinone form. In addition, four protons are transferred from the stromal to the luminal side of the membrane. By this mechanism, electron flow connecting the acceptor side of the PS2 reaction center to the donor side of the PS1 reaction center also give rise to an electrochemical potential across the membrane, due in part to the hydrogen ion concentration differences on the two sides of the membrane, and this electrochemical potential is used to power the synthesis of ATP. The cyclic electron flow through the cytochrome B and plastoquinone increase the number of protons pumped per electron beyond what could be achieved in a strictly linear sequence. Here is the explanation of this pathway, which I told you in the previous slide. It is the mechanism of electron and proton transfer in the cytochrome B6F complex. जैसे कि मैंने आपको बताया था कि प्लास्टोक्यूनोन जो है ये after getting electron, one electron will be converted into semiquinone and then into the plastohydroquinone. और मैंने आपको ये भी बताया था कि जो भी प्लास्टोक्यूनोन जो है ये इलेक्ट्रॉन जो है PS2 से लेंगे, उनके पास टू पाथवेज में वो इलेक्ट्रॉन को डिवाइड करेंगे। दो इलेक्ट्रॉन है इनके पास प्लास्टोहाइड्रोक्विनोन के पास, एक इलेक्ट्रॉन जो है वो देंगे वो साइटोक्रोम एफ को। आपको मैंने बताया था साइटोक्रोम B6F जो है इसके अंदर दो तरह के साइटोक्रोम पाई जाती हैं, बी टाइप, ये ऊपर दो तरह की बी टाइप है और एक जो है साइटोक्रोम एफ टाइप जो है। एक इलेक्ट्रॉन जो है वो साइटोक्रोम एफ रिसीव करेगा एंड थ्रू दिस आयरन सल्फर रिस्क प्रोटीन this will be transferred to the plastocyanin and to the PS1 and so on। जो आगे की सीरीज है वो इसके हिसाब से चलेगी। एक इलेक्ट्रॉन जो कि दूसरी साइटोक्रोम बी कॉम्प्लेक्स के पार्ट के पास गया है, साइटोक्रोम बी टाइप के पास गया है, ये इलेक्ट्रॉन जो है एक अनदर क्यूनोन मॉलिक्यूल को प्लास्टो सेमी क्यूनोन में कन्वर्ट करेगा। एक और पाथवे, ये है वन टर्नओवर की कहानी। एक और टर्नओवर जब आएगा, यानी कि एक इलेक्ट्रॉन जब दोबारा से ये प्लास्टोहाइड्रोक्विनोन दोबारा से इलेक्ट्रॉन की इसी तरीके से डिवीजन करेगा, तो व्हाट विल हैपन इन द सेकंड फेज, the again now you can see the state of this quinone that it is in the plasto semi quinone type. Here it was converted from quinone to semi quinone, and in this turnover this semi quinone will be converted into hydroquinone। सेकेंड टर्नओवर में क्या होगा, उसी तरीके से एक इलेक्ट्रॉन दोबारा से साइटोक्रोम एफ के पास चला जाएगा जो कि प्लास्टोसाइनिन के जरिए PS1 के पास जाएगा एंड सो ऑन चलता रहेगा और कंट्रीब्यूट करेगा टुवर्ड्स द फॉर्मेशन ऑफ NADPH। व्हाइल इन केस ऑफ दिस जो इलेक्ट्रॉन साइटोक्रोम बी के पास जाएगा, ये अगेन इस्तेमाल होगा जो पहले इलेक्ट्रॉन ने जो सेमी क्यूनोन बनाया था उसी को एक और इलेक्ट्रॉन जब मिलेगा, जो पिछली डायग्राम में भी मैंने आपको समझाया था कि किस तरीके से क्यूनोन सेमी क्यूनोन में और उसके बाद वो प्लास्टोहाइड्रोक्यूनोन में कन्वर्ट होगा। तो ये सेमी क्यूनोन एक और इलेक्ट्रॉन को रिसीव करने के बाद नाउ इट विल बी कन्वर्टेड इनटू प्लास्टोहाइड्रोक्यूनोन। अब प्लास्टोहाइड्रोक्विनोन में कन्वर्ट होने के लिए इसको 2 इलेक्ट्रॉन चाहिए और 2 प्रोटॉन चाहिए। 2 इलेक्ट्रॉन तो ये हासिल कर चुका है, लेकिन 2 प्रोटॉन जो है ये लेगा फ्रॉम द स्ट्रोमा, और स्ट्रोमा के जरिए, स्ट्रोमा से, क्योंकि ये मोबाइल प्रोटीन है, और एज द एज ऑफ द थैलाकोइड मेम्ब्रेन टुवर्ड्स द स्ट्रोमा साइड, ये यहाँ से प्रोटॉन को ले लेगा और दिस विल बी कन्वर्टेड इनटू प्लास्टोहाइड्रोक्विनोन। ये प्लास्टोहाइड्रोक्विनोन अगेन, ये जो क्विनोन का पूल है इसमें कंट्रीब्यूट करेगी और दोबारा से इस पाथवे को दोबारा से चलाएगी। अगेन एक इलेक्ट्रॉन इसके पास और एक इलेक्ट्रॉन इसके पास, लेकिन आप देख सकते हैं कि ये हाइड्रोक्विनोन जब अपने दोनों इलेक्ट्रॉन्स को डिलीवर करती है टू द प्लास्टो साइटोक्रोम B6F कॉम्प्लेक्स, तो इस दौरान में वो अपने दोनों हाइड्रोजन आयन को डिलीवर करती है टू द ल्यूमन साइड, और यही वो हाइड्रोजन आयन है जो कि एक्स्ट्रा एटीपी को जनरेट करने का बायस है थ्रू द प्रोसेस ऑफ साइक्लिक मैनर। जब हम बात करते हैं, जो कि हम आगे जाके डिस्कस करेंगे कि हाउ साइक्लिक फोटोफॉस्फोरिलेशन एडिशनल एटीपी को जो है वो जनरेट करता है। तो यहाँ पे इन दोनों की एक्सप्लेनेशन जो है वो एंड होती है। दिस इज द फर्स्ट टर्नओवर एंड दिस वन इज द सेकंड टर्नओवर। फर्स्ट टर्नओवर में सेमी क्यूनोन बनता है और सेकंड टर्नओवर में जो है वो हाइड्रोक्यूनोन बनता है। और ये बात भी काबिल गौर है कि ये हाइड्रोक्यूनोन इस टाइम पे एक अलग हाइड्रोक्यूनोन है और दिस हाइड्रोक्यूनोन इज द डिफरेंट हाइड्रोक्यूनोन। और ये हाइड्रोक्यूनोन जो है ये यहाँ पे एक पूल है पूरा, यानी कि एक ही के मॉलिक्यूल नहीं होता, काफी सारे मॉलिक्यूल्स, एक पूल है पूरा हाइड्रोक्विनोन का, क्विनोन का, जो कि फिर डिफरेंट आफ्टर रिसीविंग एंड डिलीवरिंग द इलेक्ट्रॉन टू द नेक्स्ट प्रोटीन कॉम्प्लेक्स जो है अपनी स्टेट को चेंज करते रहते हैं। This is the other explanation of the Q cycle. The transmembrane organization of the cytochrome B6F complex is shown schematically here at the quinol binding site QP. Now I'm showing, explaining this in another way कि जो डिफरेंट क्यूनोन है उनकी पार्टिसिपेशन है। सो दिस वन इज द पूल ऑफ द प्लास्टोक्यूनोन जो कि हाइड्रोजन को ले रहा है आफ्टर हैविंग द इलेक्ट्रॉन फ्रॉम द PS2, दिस विल टेक the hydrogen from the stroma and will be converted into plastoquinone, hydroquinone, and this plastohydroquinone will release its hydrogen ion to the lumen and deliver its two electron, one to the risk protein and through the cytochrome F complex and then to the plastocyanin and so on, and the other electron will be given to the quinone at the QP site. Now, through these series of cytochrome B complexes, cytochrome B type hemes, this electron will be utilized for the formation of plastosemiquinone and then again to the plastohydroquinone. This is the first turnover. In the second turnover, what will happen that again an electron will be delivered to this site and to QN site, and this semiquinone will be converted into plastohydroquinone. तो जो बात मैं आपको पिछली डायग्राम में एक्सप्लेन करना चाह रही थी कि जो प्लास्टोहाइड्रोक्विनोन फर्स्ट टाइम अपने इलेक्ट्रॉन को डिलीवर करता है वो वही वाला नहीं है जो कि साइटोक्रोम B6F के से बना हुआ है, यानी कि ये भी प्लास्टोहाइड्रोक्विनोन बना रहा है, जैसा कि इस डायग्राम में ज्यादा क्लियर है, ये भी प्लास्टोहाइड्रोक्विनोन बना रहा है एंड दिस वन इज आल्सो मेकिंग द plastohydroquinone. So a pool is developed by the activity of these two quinone, और इसकी वजह से फिर ये फर्दर ये प्रोसेस जो है, आगे का प्रोसेस सारा सेम है, लेकिन ये क्यूनन जो है ये अलग से PQH2 का पूल जनरेट कर रहा है और दिस क्यू प्लास्टोक्विनोन जो कि डायरेक्टली इलेक्ट्रॉन को ले रहा है फ्रॉम द PS2, ये अलग से इस पूल को जनरेट कर रहा है और दोनों ही हाइड्रोजन आयन को डिलीवर करने का बायस है इनटू द lumen of the thylakoid. Here are some suggested books from where the material is collected for the preparation of this lecture. Thank you very much. If you have any question or query, you can post on GCR, WhatsApp, or you can email me.